Today on the Smart Soul Hub podcast, we are going to tackle the elephant in the room. It is the paradox that every single patient points out to me when I explain this treatment. Diabetic peripheral neuropathy is caused by chronically elevated blood glucose. It damages nerves. Yet we take a syringe full of 5% dextrose, a concentrated glucose solution, inject it right next to a painful nerve, and the nerve settles down. Same molecule, completely opposite outcomes. I wanted to understand exactly how glucose destroys a nerve, so I could understand how it might heal one. And that led me to a 20-year-old paper that completely changed how I look at this therapy. This is part 5. Hi, I'm Jay Kim, podiatrist. I hold a master's in neuroscience by research. Before I was in clinic, I was studying how nerve cells die in neurodegenerative disease and developing assays in a cancer diagnostics lab. Now, I use that background to do something most podiatrists don't. Go back to the primary research and ask what's actually happening at the cellular level. This is Smart Soul Hub. This is part 5 of our series looking at the evidence behind perineural injection therapy and the use of 5% dextrose for nerve pain. Let's get into it. The paper we are looking at was published in 2003 by Tan and colleagues in the Journal of Biological Chemistry. Now, let's be totally clear up front about where this sits in the evidence hierarchy. This is a level 5 basic science study. The researchers used in vivo animal models, specifically rats and mice with induced type 1 diabetes, and in vitro models, meaning they isolated primary Schwann cells and studied them in a petri dish. There are no human clinical trials here. What this paper does is map out the exact molecular pathways of nerve damage at the cellular level. It is hypothesis generating. The researchers were trying to answer a specific question. How does prolonged high blood sugar actually cause the myelin sheath, the protective coating around the nerve, to break down? To understand what they found, I need to introduce you to the one metaphor we'll use for this episode: the cellular brake pedal. On the outside of a Schwann cell, there is a receptor called ERBB2. You can think of this receptor as an antenna that catches growth signals called neuregulins sent out by the nerve. When a Schwann cell is immature and developing, it needs these intense growth signals to tell it to multiply and wrap the nerve in myelin. But once that cell is fully mature, the rules change. Constant growth is dangerous for an adult cell. So, the body uses a structural protein called Caveolin-1 or Cav1. Cav1 acts as an inhibitor. It clamps onto that ERBB2 receptor and acts as a brake pedal. It ensures that the mature Schwann cell doesn't overreact to normal growth signals. The researchers hypothesized that chronic high blood sugar destroys this Cav1 brake. And that is exactly what they proved. In the diabetic rats, Cav1 expression plummeted to just 40 to 50% of healthy levels after 12 weeks of high blood sugar. In the cell cultures, exposing healthy Schwann cells to 30 to 40 millimolar of glucose for just 24 hours caused a massive 30 to 50% drop in the Cav1 brake. Now, I want to pause here because the researchers did something brilliant methodologically. When you dissolve a massive amount of sugar in fluid, it makes the fluid very thick. This can physically crush a cell through osmotic stress. To prove that the damage was caused by the cell actually metabolizing the glucose and not just being crushed by the physical pressure, they used fake sugars called L-glucose and mannitol. These fake sugars created the exact same physical pressure, but the cells couldn't digest them. The fake sugars did not destroy the Cav1 brake. It was definitively the biological metabolism of glucose causing the damage. So, what happens when the brake is gone? The mature Schwann cell becomes hypersensitive. The ERB2 receptors go crazy. The mature cell is suddenly flooded with intense growth signals that are only appropriate for a developing cell. The authors call this altered neurotrophism. The cell gets confused, tries to inappropriately revert to a growing state, and in doing so, it actively tears apart and destroys the very myelin sheath it is supposed to be maintaining. So, that is the mechanism of diabetic destruction. But how does this relate to perineural injection therapy? Remember, this is a level 5 basic science paper. It proves what happens in a petri dish over 24 hours. But it offers a highly plausible, speculative framework for what might be happening clinically. If chronic high glucose permanently destroys the brake, causing cellular confusion and destruction, could a short, controlled exposure to dextrose temporarily lift that same brake? If a nerve is damaged and stalled in its repair cycle, perhaps briefly lifting the Cav1 brake allows the Schwann cell to receive a temporary pulse of growth signals. It gets a quick burst of repair signaling before the glucose disperses and the brake goes right back on. This brings me to the question that is still nagging me: the threshold of time. The in vitro study showed massive Cav1 loss after 24 hours of 30 millimolar glucose. Now, 30 millimolar is incredibly close to the 25 millimolar concentration we estimate surrounds a nerve after a 5% dextrose injection. So, what constitutes chronic versus acute? Does the fluid from our injections sit around the nerve for 2 hours, 12 hours, 24 hours? If the biochemical signal from the D5W lingers too long, do we risk tipping that nerve from a brief healing state into that destructive, altered neurotrophism state? I suspect the small volumes we use clear fast enough to avoid this, but the exact duration threshold that separates a healing signal from a damaging one is a massive unknown. To wrap this up, Tan et al. Shows us that glucose acts on specific cellular brakes. Chronic exposure destroys the brake and melts the myelin. But it provides a compelling biological theory that acute exposure might just lift the brake long enough to help the nerve heal. You can read the full breakdown, complete with diagrams of the ERB2 pathway, on the blog at jkim.com.au/blog. Thanks for listening. I'll see you in part 6.